Hello, folks. We're going to talk a little bit about cells today and use a few sort of application features of cells to talk about some cell structures and cell functions. If you are following along with the assignment that goes with this, you should be able to pretty much write down the things I'm saying and doing because this is the exact assignment. All right. Why do eukaryotic cells have an internal membrane system, whereas prokaryotic cells do not? So first of all, we have to think about eukaryotic and prokaryotic cells. The biggest differences between them, well, obviously the biggest difference between them is that eukaryotic cells have a nucleus and other internal membranes, and prokaryotic cells don't. But the other big difference between them is that eukaryotic cells are pretty damn big. Prokaryotic cells are pretty small. And this is really the big key. So the question says, think about size and surface area. So size is a pretty big, obvious difference, but surface area is a little harder to think about because what it is it is all of the space completely around the cell. So one of my sort of good metaphors for this is instead of thinking of it like a cell, maybe think of it like a dining room. And so if this was a dining room instead of a cell, then you'd have a whole bunch of like tables in it and they'd all have to be served. And let's say the servers have to come in from the outside. So from where the cell membrane is, say this is the same dining room. Obviously it has less tables, but it would have an equal number of servers per table. That's not really the problem. Here's the problem is that if the servers have to come from the outside and get all the way to the inside, it's going to take them a lot longer to serve. A great big banquet hall takes a long time because they have to pick up stuff from the outside and get it all the way to the middle and all the way spread out. Whereas on the small cell, there's so much external surface compared to the inside that's pretty easy for the waiters to serve all the tables. So how does the eukaryotic cell fix that problem? Well, it fixes it by creating essentially pathways for the waiters to walk by. So if you think about the like membranes of the nucleus, but also the endoplasmic reticulum and stuff like that, this means that the waiters can more easily get out to where they have to go. They have plenty of places to put their trays and their extra dishes and grab water glasses from all of these hallways. So having the internal membrane system creates extra surface area. So the sort of easy answer to this is that a larger cell has less surface area compared to volume. An extra membrane allows for more efficient functions. So that's why it has to have that, whereas the prokaryotic cell can get away without it so much. All right, let's go to our next one. ALS affects the Golgi apparatus in nerve cells, causing symptoms such as partial paralysis and difficulty speaking. Why would these effects occur? Nerve cells communicate by sending neurotransmitters. Find out how neurotransmitters are released. Well, I can tell you a little something about neurotransmitters. Here's a nerve cell. It kind of comes all the way out to here. Here's another nerve cell. It comes around here and it's going to pick up the stuff from that nerve cell. Ignore my very badly drawn nerve cells. So how does that happen? Well, there's these little neurotransmitters and they go in between the cells. So they're traveling from one place to another. But the real question is, where did they come from? Guess what? They come from vesicles hanging out here in this nerve cell. So how do we get neurotransmitters into vesicles? Well, if you remember correctly, neurotransmitters, most of them are in fact proteins. They are produced through the protein processing system of transcription, translation in the ribosomes, release into the rough ER, travel through the smooth ER, and go to the Golgi apparatus to be packaged up and sent out. If the Golgi is not working, we don't get vesicles full of neurotransmitters and we don't get neurotransmitters. If we don't get neurotransmitters, then that stops the nerves from signaling and they are unable to move. So it stops the muscles from being able to move. The neurotransmitters cannot signal the muscles. That's how ALS works. And all of it comes down to just that tiny little Golgi apparatus with the funny name. All right, our next one. Lily pad leaves often have many large vacuoles, more than most leaves from trees or bushes. Why might this be useful? It says consider where lily pads are found. So hopefully you've encountered lily pads before, but lily pads are on water. There's my water. Just gonna have to ignore my badly drawn things. And lily pads typically float on the top of the water. So here's my lily pad leaves. They're gonna float on top of the water. Maybe I will also add to them some cute lily pad flowers. I used to canoe through a lot of lily pads. All right, so the other hint I have is think about how submarines work. That's only, of course, if you don't kind of get it. But the idea here is that what do submarines do? Well, they rise and fall in the water. You know how they work? Interestingly enough, they're real cool. They have large containers that they can fill up with water. If they fill them up with water, then they become heavier and they sink. If they let the water go, they become lighter and they float. So if you have large containers that are full of something like water or maybe even some extra air, it allows you to float. So guess what our lily pad leaves need to do? Well, they need to stay to the top of the water. So the vacuoles allow them to balance out if they are light enough and they can pick up a little water, a little sugar, and a little bit of empty space like air to let them float on top of the water. So in this case, the vacuoles, which are plant specific, allow the lily pads to float. All right. Why might a plant cell need a cellulose cell wall, but an animal cell does not? So another thing that plants have that animal cells do not is a cell wall. So it says two hints. There's two hints here. Think about the main function of a cell wall and think about the general differences between plants and animals. Cell wall is pretty easy, right? It does really kind of two things. They're both sort of structural. So the cell wall is to maintain structure, but also it's a little bit sturdier. So it maintains it in a protective manner. So it's also for protection. So why would a plant need more structure and more protection than an animal? What is really the key difference between plants and animals that isn't a symptom of them being plants and animals? Well, if you really, really think hard about it, the biggest key difference between kind of what we call a plant and kind of what we call an animal is that animals move and plants don't move. No, but really think about it. Plants don't move anywhere. They root in one place and they stay where they are, and animals move around. So if we, for example, need to go get something, we can move to get it. They cannot. They have to stay in one place. But also, if something is coming to get us, we can move somewhere. And the plants cannot, so they need more structure and more protection because they are unable to run away from danger. So that's a big part of it. As we get more into plants later in this class, we're actually going to discuss this again. So keep an eye on those differences between plants and animals because they come up a number of times. All right. Why might single-celled animals like amoeba have an intensive microfilament system and secrete a glycocalyx or ECM, which is an extracellular matrix? Okay, so let's think about each of these things separately. So first of all, make sure we kind of maybe remember what an amoeba is. It's this like blobby little guy. My pen is being real annoying today. He's very blobby. And if you think about how he moves, he's kind of like grabs and pulls and shifts himself around. So I should get a video of an amoeba moving for you, but you can go look up a video of an amoeba moving. They're actually real cool. All right, so now let's deal with each of these concepts. First is microfilaments. So microfilaments are part of the cytoskeletal system, and they're actually the smallest one. So microfilaments, intermediate filaments are the next biggest, and microtubules are the biggest. And if you remember correctly, the microfilaments include things like actin and myosin, which are part of movement. They're actually part of our muscles, but for cells like this, they just help it move. All right, so now that's part one. Part two is what's a glycocalyx or extracellular matrix feel like. Glyco in glycocalyx, by the way, means sugar. It's essentially a sticky, slimy substance that is secreted. All right, so if you're an amoeba, why would you want some real good microfilaments and secrete a sticky, slimy thing? Well, have you ever seen a slug? Yeah, same idea. First of all, you are going to need to move by pulling yourself along the ground. So it needs those filaments to be able to do that job of pulling it where it needs to go. So those are the filaments pulling it. The artwork does not get better in this class, by the way. But then also, if it can release this slimy stuff, it gives it an easier way to pull. So if it spits out some slimy stuff in front of it, then reaches out and pulls itself along, it has a way to get where it's going and something to stick to. So that's the usefulness of both of those. Sometimes these are also used to like just cluster together and stick to rocks, but they can be used like slugs to help things move around. We have cells that create and release digestive enzymes that can break up most proteins and carbohydrates. That's great. But how are these cells able to create and contain these enzymes without posing a danger to themselves? I mean, if they can just break up proteins, wait, what are cells mostly made of? Proteins. And then, of course, if they can break up all the carbohydrates, this is going to be tough. There's not going to be a whole lot left. But of our major components, proteins and carbohydrates are only 2 of the 4 macromolecules. That means the remaining macromolecules include nucleic acids. Oh, my poor mouse is just being all kinds of trouble today. Nucleic acids like DNA and RNA. And then also lipids. Okay, so if we look at those 2 things, which of those 2 are your sort of structural concept? There we go, I can get rid of this N. I mean, nucleic acids probably aren't going to interact well with enzymes. They're all rolled up over in the nucleus, or they're over giving people information. But our lipids are in fact structural. And in fact, we use lipids to separate things from other things. So if we think about our cell, so let me draw, the basic outline of our cell. When we create the proteins from our tiny little ribosomes, here's my tiny little ribosomes, the first thing we do is we dump them into the endoplasmic reticulum, which is bound by membranes. Eventually, they come down to that fancy Golgi apparatus, which releases vesicles, which are bound by membranes, so the digestive enzymes can hang out inside of membranes. And in fact, if you think about it, where do we keep things like lysosomes? Inside of membranes. So lipids are really the key to making sure that we can functionally break down proteins, but not break down every protein in a cell. That way it can release those digestive components, or the cell can pick up something, like if it's a macrophage trying to eat a bacteria, and then combine the vesicles so that it gets digested inside of a membrane, and then we can spit out the waste products. It's a really neat process. Okay, but we're biologists. Not all of our cells have internal membrane systems. That was one of the first things we said at the beginning. So what about prokaryotes? They have to also digest. They have to digest proteins. They have to digest stuff like that. They have enzymes. So how could they do this? My little hint for this one is think outside the box for this one. And it's really not any more complicated than that, which is that these guys, so our tiny prokaryotes, no internal membrane system, but they still produce proteins and enzymes, and they usually do it on or associated with their membrane because they need membranes for those things. So the little ribosomes come over here to the membrane. They produce the protein, and guess what they do? They spit the proteins right outside the cell. It's actually really neat. The digestion happens externally, and then they can uptake the broken down products and have it ready to go. So by the way, if it's ever like middle of fall and you're out where the leaves are in like a stream and you notice that they're all kind of broken down and slimy, Yeah, that's a whole bunch of like bacteria and fungi sitting on the leaves digesting them. So they're being externally digested. That's kind of cool, huh? Or kind of icky. I don't know. I like it. I'm a microbiologist. All right. I think I've only got one more. For studying cells, what are some ways you could study them to learn about which molecules and structures were involved in which jobs? And I give you the hint. Think about chemical properties and what various molecules are made from. So, this was actually a concept. We had to learn how cells worked, how they were structured, where things lived. It's not like scientists just sort of know that, or that we could always just dive into a cell and take a look. Nowadays, we can sort of look. Transmission electron microscopy is amazing, but it's still not perfect, and it just kind of gives us a vague overview of stuff. So, what are some things that we could do? Well, if we think about what stuff is made from, so we know lipids are pretty simplistic, they're just carbon and hydrogen. Carbohydrates are slightly more, they have carbon, hydrogen, and oxygen. We know the biggest difference between our macromolecules is, of course, our polar versus nonpolar. So, if we start with that, polar, non-polar, what are some of the things we could do? Well, we could split our cell up. We could actually break it apart, see which parts dissolve in oil, which parts dissolve in water, and start there. Then we can find out which parts are polar and non-polar, which is actually something biochemists do all the time. Throw it in a blender. Blender is a true biochemical tool, promise. The other thing we can then do is start to look for the things that make molecules specific. So, for example, our proteins are the only thing that contains sulfur. So if I can find a way to tag or follow sulfur, which I can make it either a radioisotope, which I have to be more careful with, or a heavy isotope by adding neutrons to it, then I could figure out where it goes in a cell, separate out certain parts of it, and follow proteins and see how they're produced, or at least see how they come in and out of a cell. And nucleic acids, they're special. They're the only ones with phosphorus. And so if you can tag phosphorus, then you can track nucleic acids. You can see them uptake phosphorus. You can see where they're concentrated with the phosphorus. And you can see if that phosphorus is doing a job. So one of the ways that we learned that DNA was genetic material and enzymes were proteins and we're able to split those up was by looking for where phosphorus and sulfur went in a cell and what jobs they did. And when we split the cell up, which ones were able to do one thing or another. So if we move the item with the phosphorus and it can replicate and make a new cell, it's genetic material, that's our nucleic acid. But if we move the item with the sulfur, it can break stuff down and do enzymatic jobs, but not replicate. That's our proteins. So it's really interesting that we can use chemical properties, so the same things we talked about earlier, to figure out where things fall in a cell and how they work together. So when you're looking for exam questions, you want to think about things like if you split cells up, how polar and nonpolar things would split out. And also if you found molecules or found segments of a cell, whether you would expect to find not just polar and nonpolar things, but sulfur, phosphorus, nitrogen, sort of the elements that are a little bit more random or go in specific macromolecules so you can match those things up. All right. That is all I have got for you on this particular study. So don't forget to write that up and submit it on Blackboard for points, and I hope you learned something great.